We are going to start a topic of pathogenesis, and in our today's lecture we will end this topic. So let's discuss about the pathogenesis of bacterial infections. So first of all, you have to understand the concept of colonization and the growth. What is colonization? Colonization is actually a multiplication of a pathogen after it has gained access to the host tissue. Okay, so means after entry or after access to the host tissue, pathogens multiply. So this multiplication or this spreading of the pathogen on the surface of the host tissue is known as actually colonization. The initial inoculum of pathogens is rarely sufficient to cause damage, so it must find appropriate nutrients and environmental conditions. Okay, so initially the infectious dose और initially जो बैक्टीरिया एंटर होते हैं उनका उनकी क्वांटिटी इज़ insufficient to cause the infection. So for disease or for infection, to spread of infection, we need many copies of बैक्टीरिया और पैथोजन. सो अगर हमें ज़्यादा कॉपीज़ चाहिए, ज़्यादा पैथोजन चाहिए, तो बैक्टीरिया मल्टीप्लाइज. सो व्हेन बैक्टीरिया मल्टीप्लाई दे नीड न्यूट्रिएंट एंड favorable environmental condition. Okay, so competition with normal flora. So competition with normal flora is also occur because normal flora is involved in the defense mechanism by covering the or by consuming the fact surface receptors. Okay, so they also compete with the normal flora and get more, they invade on the tissues by making more receptors for the available for the bacteria or pathogen. Okay, so when normal flora gets in competition with the pathogen, so usually normal flora, or if pathogen is very virulent, then uh, normal flora will be lost. And after then what is resistance? They bacteria and other pathogens, bacterial pathogens usually compete or resist our against our immune system, like bile salts, stomach acid, peristalsis, and skin secretion. So few are barriers which are involved in our immune system, maintenance of an immune system. So bacteria also resist against these immune barriers, and the bacteria usually survive against immunoglobulin A. So immunoglobulin or antibody A is actually exist in the mucus or mucosal secretions. So they escapes from the uh, IgA and then multiplies on the surface of any tissue. Another important term is bacteremia. Bacteremia is actually a pathogens usually remain localized at the site of entry, but can pass through the lymphatic vessels and eventually to the blood, where it can travel around the body. So usually presence of pathogens in the blood is actually bacteremia. So presence of bacteria in the blood is, we can say, it's a bacteremia. So it can move, move with the circulation, it can move with the it can circulate with the lymphatic vessels as well. So presence of bacteria in the blood is actually bacteremia. So bacteremia is actually different from the septicemia. Septicemia is actually a presence of bacteria and with the disease symptoms, with the appearance of the disease symptoms, is actually septicemia. But bacteremia is only presence of bacterial pathogens in the blood or lymphatics, but it is not necessary to it is not mandatory, or sometimes usually no disease or no any sign and symptom for any disease and bacteria is still present in the blood, this is actually bacteremia. Okay, so septicemia is different from bacteremia. In septicemia, disease appears, signs and symptoms will appear, but in bacteremia, no disease will appear, but bacteria will be present in the blood of a patient. Another condition is survival in the host, protection against phagocytes and complement by a capsule. So first of all, after penetration into the host tissue, bacteria need to survive on the host surface. So for the survival on the host surface, protection against phagocytes and complement is necessary. So they bacteria get protected against phagocytic cells and complement protein by the capsule. First of all, they have a capsule. The capsulated variants of bacteria are virulent, and the encapsulated remain harmless. C3b inactivation. Okay, so usually capsule is involved is the uh, escape mechanism for bacteria uh, from phagocytosis. Okay, so those bacteria which have capsule, they are more virulent, but those bacteria which don't have capsule, they are usually inactivated by C3b protein of the complement system. Okay, antigen O. What is antigen O? Antigen O is like a somatic antigen which present on the surface structure. Aggressins, leukocidin, hemolysin, these are all actually proteins which are involved in the survival of bacteria at the host surface. So another important thing is the after escape from the phagocytosis, neutralization of the acid gastric barrier by production of urease. So when we take bacteria with the food or with the anything through the mouth, then bacteria moves in the stomach. Stomach acid is very strong acid. It can kill bacteria, many bacteria, but there are some bacteria which usually produce a special alkaline enzyme. Okay, urease, like urease, one of the urease, which alkaline enzyme react with the acid of the stomach and converts this acidity of the stomach and converts the acidity of stomach is into neutral or alkaline. Okay, so when stomach acid lost their uh, acidity, okay, or when pH of stomach acid gets increased, then we can say our gastrointestinal tract is more prone toward the disease. Okay, so this is actually uh, Helicobacter pylori is a bacteria which is involved in the formation of urease. Urease react with the urease compete with this acid of stomach, and the Helicobacter survives. So urease is also a virulence factor for Helicobacter. So Helicobacter pylori causes H. pylori infection in the stomach, and ultimately, if it is remain untreated, it will lead toward the ulceration of the stomach. Then avoidance of host immune response. Bacteria is must have to avoid the host immune response. How it can avoid? Localization of the microorganism in privileged site. Okay, access to rich source of nutrients, avoid exposure to host antibodies, immune response suppression, mumps, measles. So these are actually first of all localization of the microbes in the privileged site. Bacteria prefers to remain at the site where they can usually survive for a long period of time with the presence of nutrient, or with the absence of antibodies. Okay, or with the with the absence of immune proteins as well. Okay, so usually viruses are obligate intracellular organisms, so the viruses can survive more easily in human body, because in inside our cells there are no antibodies of present, so viruses replicate inside the cells. Okay, so immune response, another important thing. Bacteria can escape, avoid the host immune response through immune response suppression. So, like mumps and measles virus stimulate T suppressor lymphocytes. So, mumps virus suppress the T lymphocytes. Okay, so T lymphocytes are a very important part of our immune system. okay, in our long term immunity. Similarly, in HIV infections, in HIV infection, they also attack our T cells, helper T cells, and these cells are also much important in our immune system or long term immunity. So if these cells gets damaged, then bacteria, it means bacteria can, or virus can, or a pathogen can avoid the our immune system. Okay, so these are actually two things, how bacteria can avoid the our host immune response, or bacteria or virus can. So in short, how microorganism can avoid our host immune response. Another important thing is the similarity between bacterial structures and the host structure. K1 antigen of the Escherichia coli and the cerebral glycopeptides serve meningitis in the neonate. So there are few structures which can be similar with the structure in the host. So similarity in the structure. Keep in mind, similarity in the structure between with the bacteria can help in the virulence of the uh, bacteria. Okay, and multitude of the serovers defined by virulence antigen. So multitude of serovers defined by virulence antigen, for example streptococcus pyogenes, rhinoviruses. ठीक है. So serovars are actually based on a difference in the types of antigen within the same species, so we can say it's actually a serotypes. Okay, so bacteria with the different serotypes or different antigen within the same species can become more virulent than antigenic variations on the viruses. Also great help also can cause great help the bacteria in the spreading of infection like influenza virus. If we talk about the virus, and influenza virus change their antigenic structure. Okay, so they alert the immune system. The previous antibodies are efficient only for the previous antigen. So our body our body is actually pro if we talk about antibodies are specific in nature, so they form against specific antigens. So if antigen structure changes inside the pathogen, then it means antibodies must have to change, or new formation of antibodies should be occur. But in our body, usually formation of new antibodies is slow or specific. So that's why in few viruses like influenza viruses, structure of antigens changes rapidly with the passage of time, but antibody structure remain old ones. So that's why influenza virus can escape easily from the antibodies or from the neutralization, and they cause infection more than one time. Survival strategies within the phagocytic cells. So bacteria, first of all, bacteria can escape from phagocytosis, but there are few strategies in which bacteria even can survive after the engulfment in the phagocytic cell. Okay, so when lysosomes forms, then they can escape from the phagosome. कैसे lies out vacuole and grow in the cytoplasm of the host cell. Okay, for example, Shigella and Listeria. So after entry into the vacuolysosome, the vacuoles become lice due to the due to enzymes production from the Shigella and the Listeria. They are common bacteria which causes this mechanism. And after the when vacuoles gets ruptured, phagolysosome vacuoles gets ruptured, then bacteria grows in the cytoplasm of the host cell. Then another strategy in which bacteria can escape from phagocytosis even after entering into the bacteria, so blocking lysosomal fusion. So after penetration in the bacteria resist the fusion of fusion which lysosome, so they escape from the lysosomal enzyme through this mechanism. Common bacteria which can cause this mechanism are Mycobacterium tuberculosis, Salmonella, commonly spread food poisoning or typhoid fever, Legionella, which cause lignarious disease. So another important thing about the toxins. What are toxins? Another virulence factor for bacteria are toxins. So there are two types of toxins: exotoxins and endotoxins. So classes of exotoxins are actually act on the extracellular matrix of connective tissues. Okay. So exotoxins, or why exotoxins are important? They can act on the extracellular matrix of the connective tissues of our host cells. There are two types of toxins, A and B. So there are two types of exotoxins, A and B. These can inhibit protein biosynthesis. Okay. So usually examples are neurotoxins as well, and they have also membrane damaging toxins, or we can say they can damage the membrane. So exotoxins acting on the extracellular matrix may cause food poisoning even in the absence of the organism. So, for example, bacteria is far away from the site of connective tissues or extracellular matrix of the connective tissue, but toxins are actually circulating in the blood. So, when they get exposed with the connective tissues, exotoxins can cause disease, like common is food poisoning from actually staphylococcus aureus or botulism, means it's a bacillus gram positive bacteria. So exotoxins, even in the absence of bacteria, they actually, exotoxins are released from first initially released from the bacteria, but if the it is present in the remain in the blood, then it can cause disease. Proteases, so they actually act as a proteases. They lyse the proteins. Proteases are actually those enzymes which can lyse the protein. Collagenases mean they can lyse the collagen. Then hyaluronidase loosen up the tissue's fiber, so they have hyaluronidase. They can act as a hyaluronidase which involve in the loosen of tissue fibers, different tissue fibers. And Staph aureus has exfoliating, means skeletal skin steam. So like for example, a specific basket staphylococcus, it has a specific enzyme or specific action from the toxins, like exfoliating. This exfoliating action helps in the skeletal skin syndrome. So two part of the exotoxins are: A part is active enzymatic part, and B part is cell binding part. So with the B part, with the B part, toxin binds with the uh, host cells. Then A part gets entry into the host cell. So after penetration, so actually A is active site, which is more important, but B, without B, A cannot act on the surface, because B will bind with the host cell, and A will act within the host cell on the surface of host cell. So like diphtheria toxin, B binds to the cell in the respiratory tract. A is in endocytosed, and it is the effector part. So A acts or cause the disease or cause the damage in the cell. Okay. So you can observe a diagram here, in which you can observe a part role of B part, which is actually only a binder, and A is actually an active component. These are toxins which can that inhibit the protein biosynthesis. It means they can not only lyse the protein, they can stop the formation of new protein. For example, Shiga toxins, a part binds to ribosomal RNA, inhibition of protein synthesis. So apart from the exotoxin of the or toxin from the Shigella binds with the ribosomal RNA of host. When they bind with the ribosomal RNA host, they interfere with the activity of the host cell. So due to limited or stoppage of activity, ribosomal RNA is unable to form new protein. So from this mechanism, By using this mechanism, Shiga toxins act as a protein inhibitors, and death of GIT tract cells occur due to low protein synthesis and poor water absorption occurs. Then it will lead toward the release of fluids from our body through GIT. This is actually a diarrhea. So Shigella toxin can cause Shiga toxin can also cause dysentery. So diarrhea is actually a loss of fluids, but dysentery is actually a loss of fluid with blood as well. So blood will be if diarrhea is with blood, then we say it will then we will say it's dysentery. Now, neurotoxins, they act as a neurotoxin because, for example, in tetanus toxins, tetanus is caused by the Clostridium tetani bacteria, so it blocks the glycine release from the neurons. Okay, blocks inhibitory neurons, muscles overactivity, and rigid paralysis occurs. So in short, we can say that tetanus can cause paralysis through inhibition of or blockage of glycine. Glycine is actually a release from our neurons, important chemicals in the as a neurotransmitter. So blockage of glycine can inhibit the stop muscles or muscles which can stop the which inhibits the activity of neurons. So overworking of neurons causes paralysis. And then other condition is botulinum. Botulinum toxins are actually released from the Clostridium botulinum. Okay, it is actually most poisonous naturally occurring toxin. It blocks acetylcholine release. So decrease. So it when it blocks the acetyl release of acetylcholine, and acetylcholine is actually a very common neurotransmitter. So stoppage of neurotransmitter cause decreased muscle contraction and flaccid paralysis can occur. In membrane damaging toxins, for example, hemolysins or phospholipases. Hemolysins cause cell lysis. For example, it may be present in Streptococcus bacteria in the form of streptolysin O. Phospholipases cleaves the lipids in membrane. For example, it is released from the Clostridium perfringens and act as a and can cause gas gangrene. So another important thing is the endotoxins. Endotoxins are all very important because they are they actually are common toxins released from the gram-negative bacteria, and gram-negative infections are very common in humans. So they are lipopolysaccharide. They exist in the form of lipopolysaccharide in the cell envelope, mostly in the gram-negative bacteria, stimulate massive release of cytokines. So when infection occurs, so when liposaccharide of bacteria enters in the body or gets in the circulation, so in response to lipopolysaccharides, many cytokines release, a huge number of cytokines release, and this huge number of cytokines uh, can cause damage to our own cells as well. So activation of coagulation can occur. So due to the more cytokines, then cause it can cause septic shock. Gram negative sepsis means endotoxins can cause shock condition or can cause septic shock, hypotension, disseminated intravascular coagulation, fever, massive organ failures, and you have already studied these stages of septic shock. Okay, so endotoxin release from the mostly from gram negative bacteria. A very important question regarding UHS. final exam, difference between exotoxin and endotoxin. You have to write all the differences in 5 marks question. Okay, so first of all, you mostly exotoxin released from the gram positive and endotoxin from the gram negative bacteria. So their endotoxins are usually exotoxins are usually present on the synthesized in the cytoplasm and may or may not be secreted, but endotoxins are component of outer membrane. So and the usually lipid endotoxin are lipid A molecules, and endotoxin are more heat stable than exotoxins, and in our innate in response assistance related to the fever, dramatic drop in blood pressure. So endotoxin are involved in the high production of cytokine. So due to increased cytokine production, innate immune through our help of immune response, shock can occur. Not every not very toxic toxicity. If we talk about the toxicity, not very toxic, small amount lead to an appropriate response that helps clear an infection. Okay, so these are all about the exotoxin and endotoxin, but keep in mind this table is very important, and you have to write it complete if we get a 5 mark question. Another third thing is damage due to immune response. Immune system antigen antibodies complex form settle in the kidney. On the another condition is the after the is there is this is another problem. Even after the antigen antibody complex formation or killing of or after recognition of antigen from our immune system, sometimes antigen antibodies complexes gets deposited on the our basement membrane, for example in the kidneys or joints. Although it can occur in some special types of bacteria, but it is a phenomena. Okay, so settle in the kidney or joints, basement membrane of kidney and joints. So on the in the kidney it's called glomerulonephritis, usually commonly from the streptococcus pyogenes. Cause it can involve in we can identify cross-reactive antibodies in this infection. So antibodies against pathogens may cross-react with the host tissue. Okay, so when they get deposited on the surface of the glomerulonephritis sorry, when the surface of the kidneys, so after the binding of complexity with the basement membrane of kidneys, our antibodies misunderstood the basement membrane, and they can act, our immune system can act on our basement membrane, can cause killing of our kidneys basement membrane. So this can move toward the cross-reactive antibody or autoimmune disease. Okay, so acute rheumatic fever, completion of throat, streptococcus infection in the throat. So now a diagrammatic representation of molecular determinants of pathogenicity. First of all, you can observe a few valence factors which are involved in the production and delivery of various factors. Attachment, then there are villi which helps in the attachment to the surface. They are replicate and evasion of immune system. Then damage to the host cells can occur. So these are actually molecules which can cause killing or attachment with the host cells. Thank you so much.